Ladies and gentlemen, welcome to Compass here live on Quotidiano TV. Today with our guest, Dr. Ernesto de Bernardis from Siracusa. Ernesto, welcome to Quotidiano TV. How are you doing?
It's going to be a very interesting experience. This is my impression, Radu.
Ladies and gentlemen, I'd like briefly to introduce Dr. Ernesto de Bernardis. He's a clinical pharmacologist at the SERT, at the Center Against Addiction in Siracusa, Italy. And in my eyes, ladies and gentlemen, Ernesto is a true pioneer in an absolute brilliant and fascinating topic related to the corona pandemic, the topic of the Tobacco Mosaic Virus. As a pioneer, he wrote 2020 an article published in Science Direct, a putative role of the Tobacco Mosaic Virus. Ernesto, welcome once more. What inspired you to look exactly and precisely at the Tobacco Mosaic Virus?
Well, Radu, like all physicians, when the epidemic started, I began to read the scientific articles as they came out, at least the ones that seemed most interesting to me. They were plenty. I was particularly struck by some observational studies that identified a lower incidence of COVID infection in smokers, which a retired colleague had shared on Facebook. I seem to remember a Chinese article and a preprint by Giovanni Landoni of San Raffaele Hospital in Milan. So I began to ponder what cigarettes and COVID could have in common. I don't know precisely why I made the connection with tobacco mosaic virus, but maybe it had something to do with the fact that both viruses were single-stranded RNA viruses, and maybe, who knows, I had absent-mindedly read something about the virus in the previous days, refreshing my memory. I searched PubMed and learned that the virus is present in the respiratory tract of smokers and that it induces the release of interferons. This was the moment. From there, I imagined that it could stimulate some kind of protective immune reaction. As you can see in the slide, on May the 9th, 2020, I shared this idea, sorry, it is in Italian, on Facebook with my colleagues, many of whom were, like me, doctors in the field of addiction medicine. No, go to the previous slide, yeah. And they got intrigued and they encouraged me to go ahead, also mentioning their direct clinical observations, which coincided with mine, of a very low incidence of COVID in our patients, who almost all are smokers. Landoni, whom I had contacted asking for his opinion, was also intrigued. And I remember that he advised me to publish my hypothesis and jokingly wrote that if it turned out to be true, I could run for the Nobel Prize. So I thought, why not? And I put together the scientific literature that could support my hypothesis, thoroughly discussed it with my wife, also a doctor in addiction medicine, and together we sent the article. medArXiv rejected it because for editorial reasons, they were not publishing hypothesis papers. Please go to the next slide. Here it is, while medical hypothesis from Elsevier accepted it right away and published it. We must say that today the finding that COVID's attack rate is lower in smokers has been confirmed by many other papers. Go to the next slide, please, Radu. Yeah, this is the 2021 meta-analysis by Simons, which shows a combined 25 to 40% reduction. Next slide. This is the published version on the Addiction Journal, and this is the revised version from the living review that Simons is still working on. As you can see in the forest plot, the protection afforded has quite a narrow curve and quite a good certainty. But I find very indicative and suggestive the study by Paleiron. Go to the next slide, please. And Paleiron was carried out in an isolated population, the military stationed on a French aircraft carrier. And smokers there show the protection from infection of 16 to 51%. And another study, go on please with the slides. This is the study by Tomaselli and co-workers from the COHEAR coordination of Catania. And this was made on the population of the small town of Troina in Sicily, where actual tobacco use and the presence of specific antibodies were verified by laboratory analysis. And here the point estimate of protection calculated from the data was at 31%. Finally, there is another very large study. Next slide, please. This is from California. And there are nearly 2.5 million persons from the USA. This is from the clinical records of all these people. It's Young Wolf 2022, which found 33 to 39% protection in active smokers. Of course, my hypothesis is not the only one. There are various other hypotheses to explain this effect. For example, some are based on nicotine. However, in clinical trials, as well as in the lab, nicotine was not demonstrated to reduce the rate of virus attack yet. There are other hypotheses. Polycyclic hydrocarbons found in smoke tar have also been considered by the Polverinos group. And in my opinion, this is a more promising avenue.
There was also, if I may interrupt you, Ernesto, a paper by Professor Son from South Korea from the late 90s, how the prion propagation in the central nervous system is inhibited by smoke with about 80%.
Very interesting.
So we know that the spike has a prion-like section, and all of you, ladies and gentlemen, may Google for scrapie, which is the prion disease, and smoke. And there you find his experiment on mice, that mice exposed to scrapie had definitely much lower susceptibility to prion disease. There are 5 mechanisms. I only kept in mind 2. Well, I'm not a scientist in this area. One is that there is a competence in the oxidative stress induced by nicotine and induced by prions. So the higher the oxidative stress induced by nicotine, the more it inhibits the prion passage. And then also neurotransmitters being loaded with nicotine do not facilitate the passage of prions to the central nervous system. But there are altogether 5. I only could briefly present 2 of the 5 mechanisms. But it seems that the smoke exposure has also some beneficial parts. But we now look, you are the pioneer in the area of the virus itself. And it looks to me like all of a sudden this virus, the tobacco mosaic virus, became interesting, like a hole would have opened in the sky and present a whole wide area of the tobacco mosaic virus, a complete unusual virus. And we know that 40% of the cigarettes contain residues of the tobacco mosaic virus. We know that smokers produce antibodies against the tobacco mosaic virus, but these antibodies differ completely from the immune reaction an animal, for instance, will produce when inoculated with the tobacco mosaic virus, as if there were two different pathways, one of immunizing against the tobacco mosaic virus by smoking, and one by inoculation. What could you tell us, please, about the antibodies which a smoker is producing, and what might be the hypothesis how an infection with the tobacco mosaic virus may look like, although it may not replicate in humans? That was our proposed second question.
I thought it was the fifth question, but I am prepared.
No problem.
Okay. I must admit I do not have enough data available. I can only record that a preprint has recently been published by a group of Brazilian researchers who in silico highlighted that certain virus surface epitopes might play a role in B lymphocyte reactivity to the virus, and I await their final publication. But I must admit I'm not enough prepared on the different reactions to tobacco mosaic virus between the aerial views and other tissues. It is, however, quite known that there might be differences in the kind of immune reactions in different tissues and in different environments. For instance, in the airways, it is frequently seen the production of IgA, while in the tissues you find other types of immunoglobulins.
Let's resume with your presentation.
Yeah.
We stopped here at the aspect of the infection presented by the nicotine and tobacco research. If we may now resume this aspect, is this slide the proper one which I choose?
Yeah, that's good. There is a well-known problem, and it's frequent that people ask, but which way the virus could reach the airways? Because indeed cigarettes burn, so the virus should be burned along with the tobacco and shouldn't survive. But really, it is only the tip of the cigarette that burns, while the rest of the tobacco cylinder is only gradually invested by the flame over the few minutes of cigarette consumption. When inhaling from the cigarette, the hot smoke passes through the tobacco cylinder and cools. The airflow invests the still intact tobacco fragments, some of which contain the virus, which is widespread in tobacco and also in other plants, generally from the genus Solanacea. The airflow can carry with it microscopic fragments of unburned tobacco and consequently also the virus particles. The inhalation of tiny tobacco fragment is supported by the finding of tobacco DNA in the bronchoalveolar fluid of smokers. Here is the paper by Busbiat in 2010. From the scientific literature, on the other hand, we know that it is possible to find the virus in the saliva and bronchoalveolar lavage fluid of smokers. Please, the next slide. This is from 1967 by Leclerc and co-workers. But next slide. This is by Fanny Balik in 2012. Tobacco mosaic virus in cigarettes and saliva of smokers. So it is evident that it must get there somehow. And it is up to us to figure out how.
But I interviewed a couple of months ago And I interviewed Professor Lomonosov, George Lomonosov from the Innis Institute, and he's an expert in the area of the Tobacco Mosaic Virus. And I also spoke to him about the history of this virus. It's funny, it's the first ever discovered virus, 1892. And I also asked him how this virus could be a future agent against, a vaccine agent against COVID. Canada is doing lots of research in this area for a future anti-COVID vaccine based on the tobacco mosaic virus. And he explained how easily this virus may encode the spike protein. I was like shocked. Look, I'm not a scientist. And then I asked him, but if this, should this go so easily, then the tobacco mosaic virus would be a perfect bioweapon. And he said, oh, yes, of course. You know, prior to that, a month before, I never heard about the tobacco mosaic virus. Like 99% of the, I guess, of the world population never heard something about the tobacco mosaic virus. And there I have a very kind expert confirming, oh, yes, of course, it may be a perfect bioweapon. And then I asked, look, but how can you destroy this virus? And he made like that. He said, only by smoking. You can only inactivate it by fire. So then I thought, okay, well, there must be some different pathways of creating immunization by smokers, because obviously by burning the tobacco, this virus residues die, and that a different route of inoculation may be somewhere hidden, or something which maybe scientists have not found out yet. But this was just a remark in addition to what you have presented so far.
Well, we find fragments of tobacco DNA in the lungs from smokers. So we know that tobacco DNA is intact. It stays in fragments of tobacco which are inhaled during the inhalation of cigarettes. Maybe filter cigarettes allow a lower inhalation rate, and cigarettes without filter allow to inhale bigger fragments. And these fragments may carry your virus, the tobacco mosaic virus. However, we don't really know, we haven't yet followed the path that the virus follows from the cigarette or the tobacco product to the body. So it might be an issue for research still to be done.
And if we could go on with the conclusions on this slide about the tobacco mosaic virus in cigarettes and saliva of smokers, what is the conclusion which scientists from Marseille have drawn?
Well, there are not many studies. However, there is an association. As I told you before, it must be studied more in depth by looking at all the ways that tobacco is used: cigarettes with filter, cigarettes without filter, non-smoked tobacco like the snus of Sweden, and other forms of use of tobacco. Each form of tobacco and each contact with plants infected with these viruses may have a different pathway to colonization and a different pathway to immunity. It is still to be studied. It will be studied in the future.
Do you think a sort of alliance of experts? Now, off the record, we will resume presenting your slides. Do you think a sort of alliance for investigating the impact of the tobacco mosaic virus on health as a sort of network between scientists would be useful for elucidating all the unknown aspects yet of this almost mysterious virus?
I'm deeply convinced that the answer is yes. And there is already a number of researchers that are performing experiments on this virus. Mostly they are doing work with kinds of derivatives of the virus, viral particles that have been changed in shape by lab procedures or by immunogenicity, because it is well known that Tobacco Mosaic Virus has been considered as an enhancer of immunity and as a carrier of different epitopes, as you said before, to prepare new kinds of vaccines. There is already a small group of researchers working on these issues, and with a quick PubMed search, you can find everything that is interesting concerning that. However, I am mainly interested in the immune-enhancing effects in the putative, because certainty is a luxury that we don't have yet. On the putative immune-enhancing effect of the intact virus from plant material.
It could have a putative role, but there might be some other immune mechanisms which might constitute even a threat to human beings. I mean, the whole area this virus covers, if I may say, is somehow unusual. It has lytic properties, so in comparison to bacteriophages, it may alter the way bacteria replicate. It may, according to its stem, either destroy bacteria or make bacteria more toxic. It may influence the cell death. It may reprogram the programmed cell death. So it plays a major role in the aging research and industry. It may also convert metal salt precursors into gold. You find on Science Direct, also and Frontiers papers on that. Now, a virus changing metal salt precursor into gold, we're in the middle in alchemy.
I don't know. It could be useful indeed.
Useful, but you have a virus covering a wide span, which is extremely wide. It may be used to reduce electricity because it may affect the boiling process of water. Its nanoparticle net on the water surface reduces the bubbles, so the energy consumption may be reduced. It may produce more efficient batteries. So I think I have enumerated like 7 or 8 ways in which this virus, obviously in addition to the immune system, plays a certain role. It can be converted into a crystal. 1946, Stanley received the Nobel Prize for having transformed the Tobacco Mosaic Virus into a 1-cube crystal. And then he resolved it, he recrystallized it 12 times. Strange thing. I mean, is it organic? Is it inorganic? What else may this virus do? It's something very unusual, and I think it's definitely a topic which needs to be investigated, and also something where a wider awareness is necessary to motivate other scientists to look at it. And I think for this is a lot of motivation and transparency necessary. But I'd like now to explain us your comparison, which you have drawn of interferon-inducing activities and antiviral effects of the TMV, tellurone, and sodium nucleonate.
Yeah. I I agree with you that tobacco mosaic virus may have some role in several fields of science. And I am particularly interested in the possible clinical role of the virus in stimulating immunity, as I told you, and I'm not that informed on other field. However, I'm deeply convinced that it may have some role both as a stimulator of innate immunity and in the specific antigen response. I'm not yet precisely sure which pathway might be more interesting clinically. As you can see here, a group of Russian researchers in 1977, first author was Balezina, found that the oral administration of tobacco mosaic virus induced the release of interferons in both experimental animals and humans, resulting in some resistance to the other viral infections. This is a fundamental type of innate immune response in the organism's resistance to COVID. Several papers show that defects in the release of interferons or in the transduction of their cellular effects predispose to COVID infection and to its severity. While the previous administration of interferons, particularly interferon lambda, or interferon-inducing drugs, please, next slide. Here, as you can see, single-chain RNA, engineered, this is an engineered RNA which stimulates immunity, at least in mice, and make them resist to COVID infection. Single-strand RNA is the same kind of RNA which you find in tobacco mosaic virus. But there are experiments also in double-stranded DNA from bacteriophages. All these immune products reduce the incidence of COVID infection. So tobacco mosaic virus could act in the same way as a stimulator of the innate arm of the immune reaction, preventing infection, and this obviously applies in subjects in whom the interferon pathway works sufficiently well, because you couldn't get a good enough response in people where the interferon pathway is damaged by genetic mutations.
So it looks that the immune impact of the tobacco mosaic virus depends also on the genetic susceptibility of the host.
Of course. Of course, is it, in example, if you cannot produce interferon enough, you cannot be primed from these kinds of immune stimulators, and they will be more or less unuseful. Instead, in an healthy subject, this should be an aid to avoid COVID infection by previous interferon release.
One of my most favorite papers, Dr. Daniel Gajdusek, whom I'm often quoting here in Compass, and I love reading, he received 1976 the Nobel Prize for a seldom disease called Kuru. Ladies and gentlemen, he published a paper called A Genetic Controlled Virus. And he was talking about a protein and about the slow virus in a way which, reading all his publications nowadays, or like a fantasy of a virus from the unorganic world, I tend to think he was maybe referring to the tobacco mosaic virus. The funny part is a tobacco mosaic virus, if there's a pathway to have this virus as a bioweapon, if the inoculation with this virus works differently than the immunization, and the interaction with human beings is according to a genetic pattern, then there is a virus with a genetic control.
Yeah, I agree.
Sorry for interrupting you, Ernesto, but I had so many questions, and
yeah.
we just...
Maybe there are too many for a normal physician. I'm not a researcher.
You are absolute ingenious with your finding, and if there would be an alliance for other physicians worldwide, maybe we'll talk off the record about it, to have such an alliance would be a way to stimulate and motivate other physicians and scientists to look at this issue and come with ideas and proposals and research to find everything which is necessary for the benefit of humanity. But please go on with the description with the stem loop RNA RAGI agonist protects against acute and chronic SARS-CoV infection in mice. If we may resume the slides, please.
Yeah, go to the next slide, please, because it is important to consider also the adaptive immunity, the adaptive immunity arm. The adaptive arm of the immune reaction might be clinically important because the tobacco mosaic virus administered in the experimental animal, as you can see, induces the formation of specific antibodies, which can also be found in smokers, tobacco chewers, and even in non-smokers, because it is widespread also in food vegetables. It can be speculated, although here the literature data are not yet sufficient, in my opinion, that some of these antibodies may be useful in counteracting the attack of the SARS-CoV-2 virus. Next slide, please, Radu. Look at this. It has been found that the human polio vaccine virus, which is an RNA virus like the COVID virus, induces antibodies against the replicase, the RNA-dependent RNA polymerase, that also inhibit the similar enzyme of the COVID virus. It could be speculated that an antibody against the tobacco mosaic virus replicase may also inhibit SARS-CoV-2 replicase. There is also a curious finding in the literature. Please, next slide. And this is by Han in 2021, the active lung microbiota landscape of COVID-19 patients. And they found that tobacco mosaic virus was present in the bronchoalveolar lavage fluid of COVID patients, but not of controls, which makes me think that the COVID virus might somehow induce the replication of tobacco mosaic virus, working like an adjuvant.
Or other way around.
Yeah, or the other way around. But in this case, maybe COVID's replicase could amplify the tobacco mosaic virus RNA. COVID could lend its RNA polymerase to the RNA of tobacco mosaic virus, amplifying the tobacco mosaic virus. Should that be the case, it is conceivable that if the organism has already developed previous immunity to the tobacco mosaic virus, its proliferation may induce an early immune and inflammatory reaction capable of setting up an hostile environment for COVID virus replication. Quite a priming of immunity. In all cases, whether innate or adaptive response, several instances of viral interference are described in the scientific literature, in which infection with a virus other than COVID virus has an inhibitory effect on subsequent inoculation of COVID. It has been seen, for example, in the experimental animal with rhinovirus and also with a strain of influenza virus. So the hypothesis that the plant virus, although not infectious, but immunogenic, may have an interference with COVID infection should not shock any researcher.
There was a paper from 2012 about the tobacco mosaic virus in HeLa cells. Ladies and gentlemen, HeLa cells are experimental cancer cells from human beings. And researchers from the Wuhan Institute of Immunology have published this paper 2012 about the way how this virus induces the endoplasmatic reticulum stress and autophagy. And what I understood from this paper, that the autophagy is a mechanism how usually a cell is processing a viral protein. The way then the cell itself gets destroyed, and by the cell destruction, the virus sequences get destroyed as well. And that surprisingly, the tobacco mosaic virus is inducing this autophagy mechanism, but this virus cannot be processed by fibrinolysis, and that these vacuoles of autophagy remain in the cells, and that on the membranes of these autophagosomes, coronaviruses replicate and polioviruses replicate. So, look, I'm not a scientist, but reading this word by word says to me that the tobacco mosaic virus has the potential of being a facilitator.
Yeah. It may interfere in several phases of the attack of other viruses, and definitely it should be studied more deeply by our scientists.
What surprise do you have here for us on this slide? I really appreciate you put a lot of effort in making this presentation for us ready. I thank you very, very much for your help.
You are welcome. I think that the study of the tobacco mosaic virus attains to the One Health approach. One Health approach is the approach that considers not only human health, but human health in comparison and together with the health of animals and the health of plants. And because we are all we humans and animals and plants and protozoa and bacteria and archaea and viruses and whatever are part of what can I say, are the same life form based on DNA, which in a way changes itself continuously. Everybody is eating everybody, and we should look at everything as a whole, as in an holistic way. And so plant viruses are important, like human viruses, like the microbiota that lives in our bodies. And it's an important and maybe sometimes forgot issue of science. However, what is this? I like sharing, and during these years I shared everything I found about the role of tobacco mosaic virus and COVID on Twitter to discuss it with other interested persons and researchers. And what can you see? What can you see on the right? I suggested, and I would have liked to see a randomized controlled trial of the effect of inhaling tobacco fragments containing tobacco mosaic virus compared with a placebo, which could be dried plant material free of viral contamination, to see if it can be protective. The primary outcome should have been the rate of COVID attack in exposed subjects, and secondarily, the duration and severity of the disease and the incidence rate of long-term complications and mortality. I had proposed these in the closing section of our publication, but no one so far has taken up this suggestion. Today, with the widespread diffusion of vaccination and the circulation of less aggressive variants like Omicron, such a study probably makes less sense. Since, however, there is no guarantee that in the future we will not have to face variants that escape vaccine-induced immunity and may be more virulent and dangerous, perhaps it would be worth pursuing this avenue as well. Using plant materials containing immunity-boosting viruses could be a low-cost approach without patent issues and available even in poorer countries. Trying it would not be difficult, and the risks would be probably very small, since our species has been peacefully coexisting with these viruses practically forever. Finally, I want to mention a curious thing that has been very intellectually stimulating for me. Many people have seen, especially during the Venetian carnivals, photos and videos of the Carnival of Venice, the masks of the plague doctors. Please go to the next slide. Here are the masks of the plague doctors. They are masks with a long hooked beak and a full face and body protection. During the plague epidemics of the 17th century, physicians and also those who dealt with plague victims wore these masks for protection. And the long hollow beak that you see was filled with dried herbs, sometimes impregnated with a medicament, teriaca, which contained at least 60 other ingredients of natural origin. The doctor would breathe the air through the beak crammed with all these strange materials. It occurred to me that this maybe was a means of administering into the incoming airflow stream plant and animal antigens, including their viruses and fragments of genetic material, which by stimulating an innate or adaptive immune reaction at the level of the lung might have made the environment hostile to the attack of the plague bacilli. Who knows?
Who knows? And who knows if it was a bacterium or not a virus? I read a paper written by Scott and Duncan, both professors in Liverpool, but I don't even know if they're still alive. And they suspect that the plague was possibly rather due to a hemorrhagic fever, also a single strain RNA virus, and not to bacteria.
Yersinia.
Exactly. This would be something very surprising, but not unexpected. I'd like you to say two more words related to this second last picture, because you've written that worldwide reports tell that tobacco smoking is less frequent in people hospitalized for COVID-19 than in respective general population. Why?
This was
as a concluding answer for our discussion.
Yeah, it's not certain yet, and research goes on, although quite slowly. And maybe we are going to know the reason once in the next few years. I'm going forward with my hypothesis, trying to to induce further research in labs which have funds and can go on. I am addiction doc, and you have you have listened to all the telephone rings of people who is looking for me, and I'm stopping at the hypothesis. I cannot go on with my forces, but it is. I would really love if anyone could take it in own hands and prove or disprove it. It could be just a wonderful thing for me.
Ernesto, thanks for having taken your time with this precious information, which is extremely motivating and interesting also for other scientists and physicians to look at this virus. And maybe the way you, and also me, because it fascinates me as well, maybe the way we spread this information would be faster than the contagious aspect of this virus. Then we may be the fortunate ones. Yeah. Thank you also for your effort having prepared these slides for us. And I don't want to go on keeping you busy, since your phone is ringing all the time and your patients are waiting for you. Thank you very much for having participated in this interview. All the best and keep in touch.
Thank you to you, Radu.
Thank you very much, Ernesto. All the best. Ladies and gentlemen, have a nice afternoon and thanks for watching Compass. Bye-bye.